In this last presentation, I will be analyzing Figure 7 as well as making concluding remarks about the paper. So after discovering that their 3A mutant was specifically impaired for the degradation of only the HMG2P and not the ERAD-L or ERAD-M substrates, the authors did a variety of control experiments in Figure 6 that altogether showed that the 3A mutation does not affect the formation of the HRD1 complex. They then went on to repeat screening of their original HRD1P mutants for specific impairment of other ERAD-M substrates. Interestingly, they found that many mutations in the transmembrane domain of HRD1P showed specificity for various ERAD-M substrates. For example, in the supplementary Figure 3, they found a mutant variant that was specifically impaired in the degradation of a membrane substrate PDR5, yet displayed wild-type degradation of HMG2P and Sec61. After this discovery, they wanted to look more into the mechanism of the recognition of HRD1P. They proposed two possible models for explaining the effect of the 3A mutation on HRD1P recognition of the HMG2P ERAD substrate. In the binding model, it was proposed that the 3A mutation inhibited binding of the ERAD-M substrate to HRD1P. Without binding, the HRD1P ring domain would not be able to recognize and ubiquitinolate the HMG2P protein, and thus it would not be targeted for proteasome degradation. In the allosteric model, it was proposed that the 3A mutation was actually on an allosteric site of the HRD1P protein that is involved in the interaction with the misfolded protein and subsequent signaling to the ring domain on HRD1P to ubiquitinolate the protein. Thus, without the signal for the ubiquitinolation to occur, the misfolded protein would not be recognized and sent to the proteasome for degradation. So here's a simple diagram I made outlining the steps they took to create Figure 7a. They transfected cells that were already expressing GFP-tagged HMG2P protein with HA-tagged wild-type or mutant HRD1P. They then allowed the cells to grow, lysed them, and spun down the ER microsomes, which would be loaded with HRD1P and HMG2P proteins within the membrane. They added a crosslinker to preserve any bonds before they added detergent to break up the microsome. And finally, after immunoprecipitating for the GFP tag, they immunoblotted for the HA tag or the GFP tag in the following figures. In panel A, you can see the crosslinker they used was DSP, and they applied it in increasing concentrations from left to right. The immunoblot for the GFP tag is the control, as this is what they had immunoprecipitated for in the first place. They also did the experiment discussed in putting a known amount of HA-tagged HRD1P protein. This allowed them to relatively quantify the amount of either the wild-type HRD1P or the 3A mutant protein that was co-immunoprecipitated with the HMG2P protein. As you can see, the ratio of wild-type HRD1P that was immunoprecipitated relative to the HRD1P input at a 80 microgram per milliliter concentration of crosslinker was the same as the 3A mutant ratio 1.34. This means that both protein variants were equally immunoprecipitated, and thus the 3A mutation does not affect binding of the HRD1P protein to HMG2P. In panel B, they show the binding of HRD1P and HMG2P is not detergent dependent, and that the 3A mutant HRD1P still shows no difference in binding from the wild type. Remember back in the introduction to ERAD-M pathway that UBC7 works with the HRD1P ubiquitin ligase to transfer a ubiquitin group to the substrate? Well, UBC7 is necessary for the ubiquitinylation to occur. The authors exploited this fact in an interesting ubiquitinylation assay where they used the lack of UBC7, indicated by the negative sign, as a negative control for no ubiquitinylation. As you can see, with the wild-type HRD1P, the smear indicates ubiquitinylation of the substrate has occurred. However, with the 3A mutant, there is no ubiquitinylation smear. This means that when the HRD1P protein was mutated, it was made non-functional for the ubiquitinylation of HMG2P protein only. Also, they showed here in panel C that the 3A mutant was able to transfer a ubiquitin to PDR5, another ERAD-M substrate. So all in all, what can we conclude from Figure 4? From A, we learned that the 3A HRD1P mutation does not affect the binding to the HMG2P protein. In B, we learned that this binding is not detergent-induced. And in C, we learned that the HRD1P 3A mutation affects its ability to ubiquitinylate only HMG2P protein, but not other ERAD-M substrates. So, because the 3A mutation did not affect binding and did not affect the overall function of HRD1P as a ubiquitin ligase, we can conclude that the recognition of ERAD-M substrates by HRD1P follows the allosteric model. So we've learned that in yeast there are 3 separate pathways called ERAD-C, ERAD-L, and ERAD-M, which together function to control quality protein folding. An ERAD-M substrate is a membrane protein that has a misfold located in its transmembrane domain. What was illuminated by Sato and colleagues in this article was that upon binding of the aberrant protein to HRD1P ubiquitin ligase, interactions occur with specific hydrophilic residues in the transmembrane domain of HRD1P. Somehow, this recognition at the transmembrane domain transmits a signal to the ring domain telling it to ubiquitinylate the substrate. In turn, the ERAD-M substrate is targeted to the proteasome for degradation. So that's the end of my presentation. Good luck on your exams!